Imagine this. If even a single cell division goes wrong, during the first 5 days after fertilization, a new human life may never develop.
But here's the most fascinating fact. Throughout these 5 days, the embryo undergoes continuous cell divisions, yet its overall size does not increase at all.
Following fertilization, the zygote undergoes cleavage. These cells compact into a solid morula. Finally, a fluid-filled blastocyst forms, ready for implantation.
Fertilization begins when a single sperm penetrates the egg. Cleavage creates a solid ball of cells called the morula. The blastocyst forms, ready for implantation. Gastrulation shapes the 3 germ layers.
In the previous part, we learned that fertilization results in the fusion of the male and female pronuclei, forming a diploid zygote. From this point onward begins the first stage of embryonic development, known as cleavage. Cleavage is a series of rapid mitotic cell divisions that begins approximately 30 hours after fertilization. During the first mitotic division, the zygote divides into 2 equal-sized cells called blastomeres. Subsequent mitotic divisions occur approximately every 12 to 24 hours, producing the 2-cell, 4-cell, 8-cell, and 16-cell stages. The most important characteristic of cleavage is that the overall size of the embryo does not increase throughout these divisions. This is because the embryo remains enclosed within the zona pellucida, a glycoprotein layer that prevents expansion. As a result, each successive division produces progressively smaller blastomeres, while the external diameter of the embryo remains almost unchanged. This unique feature distinguishes cleavage from ordinary mitotic cell division. At approximately the 8-cell stage, the embryo undergoes a crucial developmental event known as compaction. During this process, the blastomeres lose their rounded appearance and become tightly adherent to one another through adhesion molecules, particularly E-cadherin, along with the formation of tight junctions. Compaction is far more than simple cell adhesion. It establishes cell polarity and marks the beginning of the first cell fate decisions. At this stage, the embryo starts determining which cells will contribute to the future embryo and which will form the supporting extra-embryonic tissues, particularly the placenta. Therefore, compaction represents the first visible step of embryonic cell differentiation. By the 3rd to 4th day, the embryo reaches the morula stage, consisting of approximately 16 to 32 blastomeres. The term morula is derived from the Latin word morum, meaning mulberry, because under the microscope the embryo resembles a mulberry fruit. During this stage, the outer cells gradually commit to the trophoblast lineage, while the inner cells remain clustered together as the future inner cell mass. Meanwhile, the embryo continues its journey through the fallopian tube and enters the uterine cavity. Once the morula reaches the uterine cavity, Na+/K+ATPase pumps present in the outer trophoblast cells become highly active. These pumps transport sodium ions into the spaces between the cells. Water follows the sodium ions by osmosis, leading to the gradual formation of a fluid-filled cavity known as the blastocoel. This process is called cavitation or blastulation. With the formation of the blastocoel, the morula transforms into a blastocyst. A mature blastocyst consists of 3 major components. The first is the trophoblast, the outer layer of cells that will later develop into the chorion and eventually the placenta. The second is the inner cell mass, also known as the embryoblast, which gives rise to the entire embryo. The third component is the blastocoel, the fluid-filled cavity that maintains the structural integrity of the blastocyst and facilitates further embryonic development. An important concept at this stage is cell potency. The zygote and the earliest blastomeres are totipotent, meaning they have the capacity to develop into both the entire embryo and all extra-embryonic tissues, including the placenta. However, by the blastocyst stage, the cells of the inner cell mass become pluripotent stem cells. These cells can differentiate into nearly every tissue of the human body, but can no longer form the placenta. This principle forms the biological foundation of modern stem cell biology and regenerative medicine. Around day 5, the blastocyst begins to expand, causing the zona pellucida to become progressively thinner until it finally ruptures. This process is known as hatching. Hatching is essential because the trophoblast cells cannot establish direct contact with the uterine endometrium, while the zona pellucida remains intact. Only after escaping from the zona does the blastocyst become capable of implantation. Finally, between day 6 and day 7, the blastocyst begins attaching to the endometrium of the uterus. This process is known as implantation, marking the beginning of a successful pregnancy. In the next part, we will explore implantation in detail, including trophoblast differentiation, invasion of the endometrium, and the initiation of HCG secretion.
If you're preparing for MBBS, BDS, nursing, and want to master embryology through concepts rather than memorization, make sure to follow Zenvy Health. In the next part, we'll explore implantation, trophoblast differentiation, and HCG secretion in complete MBBS level detail.